Good morning, all of you. Now, today I am going to start a new topic that is biosensor from Unit First C part. Let's start biosensor and its applications. पहले समझते हैं कि सेंसर, ट्रांसड्यूसर और बायोसेंसर्स क्या होते हैं. तो सेंसर क्या है? A sensor is an object whose purpose is to detect events or changes in its environment and then provide a corresponding output. Examples are pressure sensors, ultrasonic sensor, humidity sensor, gas sensors, motion sensors, biosensors, acceleration sensors, displacement sensor, etc. तो इसमें देखिए एक बायोसेंसर है जिसे हमें डिटेल में पढ़ना है. ट्रांसड्यूसर की बात करें तो ब्रीफली यहाँ पे देख लेते हैं ट्रांसड्यूसर क्या होता है. A transducer is a device that converts one form of energy to another form. Example, pressure sensors might detect pressure, a mechanical form of energy, and convert it to electrical signals for display at a remote gauge. A sensor is a type of transducer. See next slide. Sensor characteristics. Accuracy. How much accurate reading it produces. Precision. How close is the measured value to the true value. And resolutions. Smallest incremental changes in the input that will produce a detectable change in the output. Sensitivity. Ratio in incremental output to incremental input. Selectivity and specificity depends on the unwanted signals. Non-linearity, deviations from linearity, that is deviations from best fit straight line obtained by regressions analysis. Now comes uh, point biosensor. What is biosensor? तो बायोसेंसर्स क्या होते हैं? बायोसेंसर इज एन analytical device used for the detections of an analyte that combines a biological component with a physiochemical detector. बायोसेंसर क्या है? तो बायोसेंसर्स आर analytical device which measures concentration of an analyte. And second points ये है कि बायोसेंसर्स biological materials such as enzymes, antibodies, whole cells, nucleic acid, that is DNA and RNA, is used to interact with analyte. These interactions produces a physical or chemical changes which is detected by the transducer and converted to an electrical signals. These signals is interpreted and converted to analyte concentration present in the sample. हिस्ट्री की बात करें तो प्रोफेसर लीलैंड सी क्लार्क is known as the father of the biosensor. On 15th April 1956, the biosensor that he invented was named after him as a Clark electrode. It is used for measure oxygen level in blood. In 1975, Clark's ideas became commercial reality with the successful relaunch of the Yellow Spring Instrument Company OYO glucose analyzer based on the amperometric detection of S2O2. In 1977, Carl Kaman introduced the term biosensor. सैंपल्स में हम क्या क्या सैंपल्स लेते हैं? वी सी हियर environmental samples such as air, water, soil, vegetation. Food samples हैं, human samples such as blood, urine, saliva. Then cell cultures developed from where we can take as a sample. Now transducer ट्रांसड्यूसर में क्या है, बायो रिसेप्टर्स हैं, इंजाइम्स, एंटीबॉडीज, सेल्स एंड न्यूक्लिक एसिड्स हैं। एंड नेक्स्ट है एफईटी डिवाइसेस, मतलब फील्ड इफेक्ट ट्रांजिस्टर। देन है न्यू नैनो वायर एरे, नैनो पार्टिकल्स, इलेक्ट्रोड्स एंड इलेक्ट्रिकल इंटरफेस। तो ये किस तरह से सिग्नल जो एम्पलीफाई होते हैं प्रोसेसर के पास जाते हैं, प्रोसेसर के बाद ये जो है अपने रिजल्ट को स्क्रीन पे डिस्प्ले करते हैं। इसी को हम डिटेल में वर्किंग में समझते हैं। देखिए हाउ बायोसेंसर्स वर्क। हियर यू कैन सी substrate to be detected passes through a thin outer membrane and it is detected by recognition system, immobilize enzymes, antibodies or cells. Then interactions of substrates and immobilize detectors which produce products. Products produce electrical signals or chemicals, gas or heat generation. Uh, then transducer converted the product level into conventional electrical signals. They reached at amplifier, amplifies the electrical signals and then it displayed on screen read out output signals. तो यहाँ पे हम देखते हैं पहले कुछ इसके टर्म्स हैं वी शुड बी नो। एनालाइट, एनालाइट क्या है? सच एज ए ग्लूकोज, लैक्टोज एंड कोलेस्ट्रॉल। बायोलॉजिकल कंपोनेंट्स कौन कौन से हैं? जैसे एंजाइम्स हैं, सेल्स हैं। ये एनालाइट बायोलॉजिकल कंपोनेंट से बाइंड अप करते हैं। कहाँ पे बाइंड अप करते हैं? ये ट्रांसड्यूसर के पास। ट्रांसड्यूसर क्या करता है? इसे एम्पलीफाई करता है, इसे इलेक्ट्रिकल सिग्नल में कन्वर्ट करता है, देन ये प्रोसेसर के पास भेजता है। प्रोसेसर क्या करता है? इस सिग्नल को जो है स्क्रीन पे या डिस्प्ले पर ये कंसंट्रेशन के फॉर्म में दिखाता है। तो वी कैन से बायोसेंसर्स आर एनालिटिकल डिवाइसेस विच मेजर concentration of an analyte. Okay, we can see here working of biosensor such as biosensor is an analytical device which employs a biological materials to specifically interact with an analyte. Analyte क्या होते हैं? तो अभी हमने आपको बताया these are such as glucose, lactose. These analyte interact and produce some detectable physical changes that is measured and converted to an electrical signals by a transducer. अब ट्रांसड्यूसर क्या होते हैं? ये क्या करते हैं? इलेक्ट्रिकल सिग्नल में कन्वर्ट करते हैं। Now finally the electrical signal is amplified, interpreted and displayed as an analyte concentration in the solution or preparation. A sensor that integrates a biological element with a physiochemical transducer to produce an electronic signals proportional to a single analyte which is then conveyed to a detector. तो इस तरह से ये डिटेक्टर के पास पहुंच जाता है। यहाँ पे ट्रांसड्यूसर है क्या? एक स्टिफ फॉर्म है, एक स्टिफ फॉर्म में है जिस पे कि एनालाइट और जो बायोलॉजिकल कंपोनेंट्स है ये दोनों इंटैक्ट करते हैं। तो इन ट्रांसड्यूसर का जो वर्क है, फंक्शन है, वो ये है कि इंटरेक्शंस और एब्जॉर्प्शंस ऑफ एनालाइट and biological component on surface of transducer and it produces physical or chemical, we can say physiochemical changes this detected by transducer. This is converted into electrical signals by transducer. Now this amplified by amplifier and amplified by processor and converted into analyte concentration present in the solution, and then it is displayed on the screen of biosensor. तो ये बायोसेंसर की वर्किंग है. नाउ यहाँ पे देख सकते हैं हम बायोसेंसर्स को किस तरह से क्लासिफाइड किया गया है ट्रांसड्यूसिंग एलिमेंट में और बायो रिकॉग्निशन एलिमेंट से. जो ट्रांसड्यूसिंग एलिमेंट है ये मास बेस्ड बायोसेंसर है, देन इलेक्ट्रोकेमिकल बायोसेंसर है, देन ऑप्टिकल बायोसेंसर है. ये मास बेस्ड बायोसेंसर फिर फर्दर डिवाइडेड है मैग्नेटो इलेक्ट्रिक एंड फिजो इलेक्ट्रिक. फिजो इलेक्ट्रिक को फिर दो भागों में बांटा गया है, क्वार्ट्ज क्रिस्टल्स, माइक्रोबैलेंस, देन सरफेस acoustic wave. देन इलेक्ट्रोकेमिकल बायोसेंसर्स में है पोटेंशियोमेट्रिक सेंसर, एम्पेरोमेट्रिक सेंसर, इम्पेडमिक इम्पेडमेट्रिक सेंसर एंड कंडक्टोमेट्रिक सेंसर. ऑप्टिकल बायोसेंसर की बात करें तो सरफेस प्लाज्मोन रेजोनेंस है, रमन एंड FTIR है, फाइबर ऑप्टिक्स है एंड अदर्स है. बायो रिकॉग्निशन एलिमेंट्स में एंटीबॉडीज हैं, फेस हैं, DNA, इंजाइम्स, बायोमेट्रिक्स. नाउ सेंसिंग एलिमेंट्स. फर्स्ट वन इज इंजाइम्स. इंजाइम्स क्या होते हैं? तो इंजाइम्स आर प्रोटीन्स विच हाई कैटालिटिक एक्टिविटी एंड selectivity towards substrates. They have been used for decades to assay the concentration of diverse analysis. Their commercial availability at high purity levels makes them very attractive for mass production of enzyme sensor. Enzymes have been immobilized at the surface by the various methods such as by adsorption, covalent attachment, entrapment in a gel or an electrochemically generated polymer, in bilipid membrane, or in solution behind a selective membrane. Enzymes are commonly coupled to electrochemicals and fiber optic transducer. Now next देखते हैं एंटीबॉडीज. Antibodies are proteins that show outstanding selectivity. Molecules larger than about 10 kilodaltons can stimulate an immune response. Many antibodies are commercially available and commonly used in immunoassay. Antibodies are usually immobilized on the surface of the transducer by covalent attachment, by conjugation of functional groups such as amino, carboxyl, aldehyde or sulfhydryl groups. The surface of the transducer must be previously functionalized with an amino, carboxyl, hydroxyl or other group. So these enzymes and antibodies are immobilized by various methods. Now next, microbes. The use of microorganisms as a biological elements in biosensor is based on the measurement of their metabolism. Microbial cells have the advantage of being cheaper than enzymes or antibodies, can be more stable and can carry out several complex reactions involving enzymes and cofactors. Conversely, they are less selective than enzymes. They have a longer response and recovery times and may require more frequent calibrations. Now, see here, transducing elements, principles of detections, physioelectrics, which measures changes in mass, electromechanicals, measures changes in electric distribution, optical measures changes in light intensity, and calometric, which measures changes in heat. Now see piezoelectric, in which materials comes quarters tourmaline, rosely salt, lithium sulfate, barium titanate. Shows piezoelectric effects, electric polarization produced by mechanical strain in the crystal, and others. Amplifier. An amplifier is an electronic device that increases the power of a signal. Power amplifier, transistors amplifier, operational amplifier, etc. Now A oblique D, analog to digital converter, counter controlled A by D converter, successive approximations, analog to digital converter, flash or parallel, uh, analytical to digital converter, then dual slope A oblique D converter. Now applications: glucose monitoring, pregnancy test in diabetes patient, historical market driver, and in environmental application, example detection of pesticide and river water contaminants such as heavy metals and which are present in waters or in our environment, and remote sensing of airborne bacteria in bioterrorist activities, remote sensing of water quality in coastal waters by describing online different aspects of clam ethology and biological rhythms, growth rates, spawning or death records in group of abandoned bivalves around the world, and determining the level of toxic substances before and after mediations, routine analytical measurement of folic acid, biotin, vitamin B12, and pantothenic acid as an alternative to microbiological assays, and applications are in drug discovery and evolutions of biological activity of new compounds, and detections of toxic metabolites such as mycotoxins. So these are the applications of biosensors: glucose monitoring in blood, in agriculture, food industries, detections of viral, fungal, bacterial diseases of plants, detections of food total microbes in soft drinks, determine freshness of fruit, meat, fish, etc. etc. These are applications in agriculture and food industry. Now, disadvantage: heat sterilization is not possible because of denaturation of biological materials. Stability of biological material such as enzymes, cells, antibodies, tissue, etc. depends on the natural properties of the molecule that can be denaturalized under environmental conditions such as the pH, temperature, or ions. And next, the cells in the biosensors can become intoxicated by other molecules that are capable of diffusing through the membrane, and fabrication is costly. So these are the disadvantages of biosensor. Now, conclusions. For biosensors, wireless biosensors networks, we can see that developing a biosensor is related with the interdisciplinary study as a biology, biochemistry, and physics, kinetics, and mass transfers. Knowledge is required for this study and proportional to the technological development and increase interdisciplinary studies. Biosensors as being more useful and having more uses area day by day. Recent development topics which are electrochemical biosensor, fiber optic biosensors, carbon nanotubes, protein engineering. So in near future, the fabrication and use of biosensors is going to be revolutionary. Now, thank you all of you